start. All right, well, our presenter this afternoon for this mini session is Ted Harris. He's a research professor working for the Kansas Biological Survey, or KBS, at the University of Kansas. He holds a PhD in ecology and evolutionary biology from KU. Ted studies cyanobacteria, blue-green algae, and just about every respect imaginable, with particular interest in harmful algal blooms, or HABs. So with that, Ted, take us away.
Thank you so much, TJ. I'm so glad to be here today. Even though it's virtual, I'd love to be in person, but obviously that is not the case this year. So I want to talk a little bit about these blue-green algal blooms that we've seen. And of course, you can see these in a couple of your pictures here to start with. And you also see that there are some other tubes filled with mud, and we will get to what that means and how these things work together. So just a little presentation roadmap of where we'll go. We'll quickly cover blooms and consequences. I think most are familiar, but we'll just quickly go through those 3 to 5 minutes, something like that. And then we'll spend sort of the last 10 minutes of the talk talking about how we find long-term trends over time, why we would look at those, why lakes are changing if they are over long time scales. And then we'll sort of sum up with how does all this past inform our present and future. And then we'll have a quick summary slide. So first, these harmful algal blooms, or HABs, H-A-B-S, in Kansas, are exclusively made of blue-green algae. This blue-green algae is technically bacteria, cyan, O bacteria. Cyan is the blue-green color, if you remember. So blue-green algae, blue-green bacteria. We call them algae because they look like algae, they function like algae, they compete with all of the real algae, and so we think of them as algae. These blue-green algae are different from others, though, in that they can produce potent toxins, one of which is even more potent than cobra venom. It is unbelievable how potent some of these toxins are. They rank among one of the most toxic producing organisms on the planet, which is pretty impressive for something that is so microscopically small. These toxins, unfortunately, have poisoned dogs, cattle, have caused human illness, and so they ultimately result in recreational closures at our lakes because we don't want people or animals coming in contact with those toxins. They can also produce taste and odor compounds, and these taste and odor compounds can double the cost of providing finished drinking water to the consumers. So if you're someone who gets water from a lake that is experiencing these blooms, you can probably rely on the fact that the cost of getting that water, that treated finished drinking water, will increase if these blooms are there. They don't always produce the toxins or the taste and odor compounds, but we don't really know why, and they're very difficult to predict when a bloom will be toxic or taste and odor producing and when it will not. These blooms are not just confined to large reservoirs like Milford, the largest in the state. Here you can see the bloom in Military Marina Cove. You can also see just how thick that bloom can get. So this bottle is about 3 inches tall, which means that scum is about 2 inches thick. You can see the top part is a little bit more living than the bottom part, but still, this is part of the issue. You can see it also affects smaller lakes, like Lake Shawnee, and other federal reservoirs like Sebelius, Webster in the west, Canopolis in the central part, and even Marion. This is a picture from 2021. This summer in early June, about 90% of the lake surface looked like this when we showed up and drove around that day. So these blooms have a big impact in small locations, large locations, and they occur in, of course, lakes that are large, small, medium-sized, all over the state. To kind of put that in perspective, I've gathered a lot of KDHE's response data. In this system, there are a response that they hear from. So if someone calls and says, hey, there looks like there's some gross scum on Milford, they would go to Milford and collect a sample. The pie charts represent two things. One, the size of that pie chart represents the number of times that KDHE has been there or been called to a site or had or knew about an issue with blue-green algae. The colors represent what was done with those samples. So progressively a watch, warning, and then hazard in red. That means that the bloom is worse and worse. And of course, right off the dot, you can see that there are dots all over the place from where blooms may have been observed or were actually found with these colors. And you can also pick out Milford, Marion, Webster, some of these lakes that unfortunately we have seen a lot of blooms in within the last decade. So why do they occur? I won't focus a lot on this today, but in really basic terms, they are just tiny plants. And so plants need light, nutrients, and water. These tiny plants are already in water. They've got that covered. And so in particular, they need light and really nutrients. Just like when you add nutrients to the terrestrial landscape, you get more yield out of crops. Well, when you add more nutrients to water, you get the same thing. And we'll see that particularly phosphorus, nitrogen and phosphorus are very important, but particularly phosphorus. And I have an example that shows that. These are not the only factors. Temperature, wind, the season, all these things underpin how HABs form and whether or not they are toxic in a very complex puzzle that's been tough to unravel. So where can we find answers to these issues? And one place we can find answers is the sediment. And really, this starts this talk, and we're sort of changing gears from what we sort of know about them to now what the past can tell us about the future. So we really want to see what were the long-term signals coming in to where we are now. And so these sediment cores, which I'll show and talk about next slide, are historical reconstruction of what's happened over time with water quality using the sediments. This includes the HAB events, and it forms as an integrated timeline of these events. The other way is, of course, to gather as much historical data as you possibly can from different agencies, different time points, anything that can help give us an idea. We use both of these in concert to help us determine what is happening in the long term when it comes to these harmful algal blooms. The sediment coring can be simply put like rings on a tree. So just like there are tree rings that show us the number of years starting with when that tree was first grown to now later, you can picture sticking in a tube and pushing it all the way down to the core of that tree. You would get one little slice of each of these layers. The lake sediment is similar, where we push these tubes down into the sediment and we go from the impoundment of that reservoir or lake when it started to be a lake, all the way to these recent years. So that the oldest sediment is down deepest and then the newest sediment is here. And again, these layer over time to give us this integrated record. Here's a picture of myself and Professor DeNoyles collecting a sediment core on Milford. So one question would be, why the cores and the data? They both seem to give a long lake story. So why both? And I'll make an analogy with a book. The sediment cores are sort of like a Cliff Note version of the book. For those of you that are younger, a Cliff Note version is essentially a summary, a short summary of an entire book. It's how those people used to cheat back in the day before there was internet on their book reading assignments. So it's really low resolution, but it's got a full view. It's got all the important events. It tells you the full story, but it's not going to include minor characters, minor events, things that really aren't that important. So not very clear on when exactly everything may happen, but everything is there. Alternatively, in the same analogy, each discrete data point, all of this data we've collected over time, is like an individual page in a book. So for example, here we see SECI, a measure of water transparency, on a lake at Canopolis. You can see most years they've only been one time. This actually is KDHE data, I believe. And there are whole years, right? 3-year spans where there is just no data. So these are like these individual pages in that they're a really high resolution. You can read exactly the words on that, but they have a very limited view. And because these samples are taken way less than 1% of all the days, these pages may or may not tell the full story. So here, of course, we have our Wizard of Oz example. And in this case, looking at these samples, we were happen to start and end. And because maybe we know something about the story, we know what all of these pictures mean in between. If you don't know the story, then these pictures look funky and you're not quite sure how Dorothy gets home again or what happens in general. So without those cliff notes or the sediment core, the story really isn't that clear. It's hard to make sense of it, or at least a lot more difficult. But with the cliff notes, all of a sudden these pages that we had ripped out of that book all of a sudden make sense. So the pages add to the story instead of trying to tell the story alone. We know the full story, and now we have added sort of data and information. And so when those two things come together, they give us two lenses to look at the past and how and why things have changed. So now we're going to switch to the results. What I'd like to do on this slide is orientate you to the slide. And then the second one, we'll talk about what the sort of colors in these things mean. Each one of these graphs is where we have collected one of these long-term sediment cores on these lakes. And how each of these works is this is blue-green algae, BGA. And it's not as important to know these numbers on this y-axis. What is important to know is that 0 means there's not a lot of blooms. And as this increases along this axis and the numbers get higher, there's more and more blooms. And then this, of course, is over time. You'll note that some of the cores we were not able to specifically date or estimate a year, but still deeper sediments mean deeper back in time. So hopefully that gives you some orientation to this. And now what I'm going to do is I'm going to put these black arrows on to talk about the change quickly in each of these lakes. First off, I'll go out west here to Sebelius and Webster. So here we see Sebelius and Webster. They have a yellow box around them. And the reason is because we've seen a short-term change in these systems. 5 or 10 years is where we started to detect this change. So in Sebelius, it's clear right here, 2005, somewhere in there, later 2000s, we start to see this little increase here. Still over time, you know, the average to look at here over time is a pretty straight line, but we do see this little short-term increase here and the same thing in Webster. Unfortunately, in many of the other lakes, particularly as we go east, we see that there are long-term changes. And so look at how much longer these arrows are, indicating where these periods of the most change have been. So we're in the middle of wrapping up Lovewell and Perry. Those reports haven't come out yet. They will within a month. But unfortunately, in these two systems, what we saw is a dramatic increase from nearly impoundment or when that system became a lake until virtually now. Some of them, like Lovewell, does have this nice decrease that we don't know exactly what that means yet. We're working on that now, but still there is a long period of increasing change in the amount of blooms. In Canopolis, we see that true. From about 1960 to 2000, it increased in blooms. We knew there were some at the start. Those were documented, but still we see this progressive increase. And then unfortunately, as we go east to some of our larger reservoirs like Marion, Perry, and Milford, for example, in Milford, we see clearly that this arrow just keeps extending. Blooms just keep getting worse and worse in Milford. Blooms keep getting worse and worse in Marion. And blooms keep getting worse and worse in Perry over time. So all three of these lakes show this trend that just keeps going. These are some of our largest reservoirs in the state. Of course, this is highly concerning. So of course, the question that you're asking now is why? Well, why are they increasing? And I will go over two of the factors, although there are many more that we are looking into. The first, which most of you probably heard about and even talked about and hopefully engaged some of the speakers in today, is phosphorus, or P, this increase in phosphorus. Here, I'll show a Milford example. It's one of the clearest examples that we have. So the top part of the screen here is, of course, these cores that we collected from Milford. This is actually where we're able to date one of them near Wakefield that we took. And that gives us a very clear view that for many years, there were not many blue-green algae in Milford until about the year 2000. And then all of a sudden, as you can see, the blue-green algae starts to increase. The signal starts to note. And of course, here we dated this in 2017. This was one of those really bad bloom years where you see this scum totally piling up in Military Marina. So then when we look at how the data was collected, for example, here's somebody in my lab, Javier Morales, collecting a sample where you just dunk this bottle down in the water, and then all of a sudden you can analyze it for nutrients and chlorophyll and all sorts of other things. Again, it gets this part of the water right there at that day. It's very clear, but it lacks what's happening over the course of the reservoir as a whole. Still, when we get all of this data involved. So here's the amount of phosphorus. Again, increasing is worse. That means there could be more algal growth. And here we are over time. What you can see is around 2000, not surprisingly, we see a big increase in phosphorus that's concurrent with the blooms. And so this really linked the problem to what is causing it, one of those primary factors. We saw similar things in Marion, Canopolis, and somewhat in Perry and Lovewell. So we do have some reservoirs where the nutrients appear to be a good indicator. In others, they are not. One question would be though, okay, we know there's more phosphorus, but where is it coming from? And so I have a little schematic here where we have the inflow, and we also have phosphorus coming from the bottom of the lake. As you increase the amount of phosphorus coming into a lake, what happens is, of course, you get algal blooms, but then they have a positive feedback loop where the oxygen near the bottom actually goes away under some circumstances. And then when that happens, phosphorus actually loads from the lake. It comes off the bottom and it gets back up into surface waters. That's called internal loading. The lake is internally pumping in more nutrients that are just not coming in. Unfortunately, the more nutrients that come in, usually we find the more nutrients get released from the sediment. So it's sort of this double bad process. And in Milford, right, even though we know the problem, one question is how much is coming from land use changes over time and how much is coming from the lake itself. My group's looking into that in Marion right now to help rectify that, but still there are these questions of, well, where and how exactly do we fix that? So some other changes that really favor these blue-green algae, there are specific things in the climate. Of course, there's temperature, as I said, but on this one, I'm actually going to focus on wind. So in this case on the left, I want to give you two schematics, one where there is gentle wind or very calm conditions, and one where there is strong wind. These blue-green algae have the ability to become buoyant, and so they float near the surface, just like that picture that you saw on Marion. And this is why we get lake-wide blooms, particularly when there's not a lot of wind. Additionally, it concentrates all of it near the surface, which is where you interact with it. However, when there's really strong winds, the algae actually get mixed throughout that whole water column, this whole area, sort of here vertical. So they get diluted, as well as their toxins get diluted by mixing it down deep. And also blue-green algae hate getting mixed deep. That's the one thing they really, really don't like compared to the other algae. To sort of link this with what's happening, a long-term wind study out of Kansas State found that average wind is staying constant. So it's not really changing, but there's an increase in the number of calm days, and there's an increase in the number of strong wind days that offset to keep the average the same. This increase in calm days makes perfect conditions for blooms and allows them to form and thrive. Strong wind days actually, though, makes it harder for the blooms to form. And so what's happening is these changes are making the blooms more dynamic, more transient, and much harder to predict. But what does this all mean for the past coming into the present and future? So what we know now is that blooms will continue to increase in occurrence and magnitude in a number of our largest lakes, particularly seemingly in eastern Kansas. We don't know if that's true yet. We're still coring more lakes to find out how much this east to west trend held, but that is what we see and project. So what does that mean? Well, more blooms, of course, means more expensive, advanced treatment for drinking water. Those costs get passed on to you. It also means more HAB warnings and closures at the recreational lakes that you know and love and want to go fishing at and want to play in, myself included. It also means that there's more health risks for animals and humans, of course, that live around these reservoirs because of these potent toxins that can be produced by the blooms. And it even means smaller fish because these blooms actually disrupt the lake food web and sort of limit the amount of nutrients that go to a lot of our sport fish, like largemouth bass and even sunfish that are sport fish. So there are all sorts of changes that these blooms will have over time. To sum up this talk, blue-green algae have increased in many of our large lakes. We cored Tuttle and Wakanda this year, two of our larger lakes, to see what's going on in those lakes which have not had any KDHE watches. Will they eventually, or is there no sort of trend there? We're finding out right now. The cores and the data together give these two lenses that allows us to focus in on when and why we've seen more blooms or why blooms will go away. We're hoping we find lakes where blooms have gone down over time to help understand why that's happening, because that is, of course, where we want to go. The blooms are increasing because of changes in the nutrients and the climate. It's important to remember that we can control the long-term nutrient loading to our systems. That's the part we can control. The climate is a lot harder. And unfortunately, these climate shifts seem to be helping the blue-green algae, at least sometimes. And unfortunately, it does not look like those times are beneficial for us. And ultimately, more blooms means that there are higher drinking water costs because it takes more and expensive processes to treat them, particularly for Kansas River Basin residents and those that live in the Kansas City area. You either get your water from a reservoir or it goes through that reservoir into the Kansas River, where then you get your water from. With that, I would really like to thank everybody who stuck around here and who did not go to lunch early. We are working, as I said, on a webinar with Perry and Lovewell right now with the water office. No date has been set yet, but that will probably be sometime in the next month, and we'll go more specifically on why we see the changes in Perry and Lovewell. So thank you, and I'm happy to take any questions in the 45 seconds that we have left.
I just say we just have a few seconds here left. I think Chris Jensen threw one in the chat there. Yeah.
External. Yes. So Chris, that is an excellent question. We hope the answer is yes. There may be a time lag though. And now all of these nutrients that have gone in, it may take some time, maybe even as long as a decade, or in some systems we found it takes even longer, essentially for all of that phosphorus to become buried so it can internally load or get flushed out. So yes, it will ultimately make a big difference. In the short term, it may not be noticeable.